Genetic modification of proteins. Protein genetic modification involves altering the genetic code of an organism to produce proteins with specific, enhanced, or novel properties. This process is crucial in various fields, including biotechnology, pharmaceuticals, and research. Here's a general overview of the key steps involved. 1. Gene cloning. To obtain a particular enzyme of interest, the gene encoding the enzyme is first cloned. This process allows for the heterologous overexpression and production of the enzyme in sufficient quantities for practical use. Cloning a gene involves several steps, beginning with the template DNA, which contains the gene of interest. This gene is typically amplified by PCR and inserted into a vector. The vector plays a crucial role in gene expression, as it contains the necessary elements to control when and how the gene is induced and expressed, determining the production period of the protein. There are different methods to clone a gene into a vector. One common method involves joining the gene to the vector using a ligase enzyme. Alternatively, ligation-independent cloning (LIC) methods can be employed, which will be discussed later. Regardless of the method used, it is essential to sequence the resulting construct. Sequencing ensures that the gene of interest is correctly inserted into the vector and that no unwanted mutations are present. 2. Directed site mutagenesis. To obtain genetically modified proteins, we use a thermal cycler to amplify the gene of interest with primers designed to introduce specific mutations. These primers, modified by a polymerase, facilitate the creation of a construct containing the gene with the desired changes. The process involves performing an amplification reaction using the gene of interest as a template in the thermal cycler. This approach results in the generation of the desired construct with the mutated gene. 3. Protein production. Once the gene has been cloned, the next step is the hyperexpression phase for protein production. First, a pre-inoculum is grown. The recombinant bacterial culture is then cultivated in E. coli media at 37 degrees Celsius until it reaches the induction stage. This is used to inoculate a larger culture. Once the desired growth phase is reached, gene expression is induced by adding the inducer. The induction process depends on the vector used in cloning, specifically the promoter it contains. The inducer can be a T-boosting agent or a molecule that binds to the promoter, such as IPTG, a lactose analog. Additionally, the culture can be induced at different growth phases: lag, exponential, or stationary phase. After induction, the culture can be grown for varying durations and at different temperatures. After the induction period, we harvest the cells, which are collected by centrifugation and then disrupted by sonication or using a French press. Then, the lysate is centrifuged to separate the supernatant with soluble proteins. The supernatant is brought into contact with the resin to initiate the protein purification process via affinity chromatography. In this case, a cobalt resin is used because the gene cloning introduced a sequence encoding a his tag at the amino or carboxyl terminus of the proteins. This his tag allows for purification using immobilized metal affinity chromatography, IMAC. The protein of interest is then deposited on a column and eluted with a 150 millimolar concentration of imidazole. The eluted protein is observed by SDS page electrophoresis. Now, we will look at different examples of genetically modified proteins. Different proteins, such as the enzyme LP3485 galactosidase, can be genetically modified. This enzyme was cloned into the Puri3-CETA vector using the LIC cloning method illustrated in the diagram. This method consists of two main steps, facilitated by the design of reaction-specific primers. These primers have one region that hybridizes to the gene of interest and another region that hybridizes to the vector and contains a his tag sequence. The first step is a PCR to amplify the gene. The second step involves an amplification where the DNA strands of the gene serve as primers, and the template is the vector. Once the construct was obtained, production was carried out. The culture was grown to the exponential phase and induced with IPTG for 18 hours at 22 degrees Celsius. Cells were collected by centrifugation and disrupted using a French press. The protein was purified by affinity chromatography using a cobalt resin, owing to the His tag inserted at the carboxyl terminus. The protein was eluted with a concentration 150 millimolar of imidazole and then dialyzed to remove the imidazole. The galactosidase activity of the protein was assayed using both soluble and immobilized protein. This enzyme exhibits galactosidase activity, but it also has transglycosidase activity. It has been observed to produce galactooligosaccharides and heterooligosaccharides. To enhance this synthesis capacity, specific residues of the enzyme can be mutated. For instance, residues in the -1 subsite can be mutated to favor the synthesis reaction over the hydrolysis reaction. An initial step could involve constructing the N3421TW553F mutant, which has been previously developed for a galactosidase from the GH36 family of Geobacillus styrothermophilus. Based on the results, additional mutants can be designed to further improve transglycosidase activity. Additionally, the nucleophilic residue, D486, can be mutated to create a glycosynthase, an enzyme that loses its hydrolytic activity and functions solely in synthesis. Glycosyntheses typically have a broader substrate spectrum than their corresponding glycosyl hydrolases, so their synthetic capacity should be evaluated using a variety of donor and acceptor substrates. Finally, another enzyme we have worked with is the lipase from Geobacillus thermocatenulatus. We used a mutant of this enzyme in which the native cysteines have been removed. From this mutant, we created several variants by introducing cysteine residues in different regions of the enzyme. These variants will be used as starting points for various chemical modifications. The enzyme is produced by inducing the culture at 42 degrees Celsius once it reaches the exponential growth phase. After 18 hours of induction, the cells are collected by centrifugation and disrupted using a French press. To purify the enzyme, it is adsorbed onto a butyl-cephalose support, a technique that allows for selective and reversible adsorption of lipases while separating them from other proteins. In conclusion, obtaining and enhancing enzymes involves several crucial steps. Initially, the gene encoding the enzyme is cloned and inserted into a vector for controlled expression. The gene is then hyperexpressed in host cells, which are induced using agents like IPTG. Post-induction, the cells are lysed, and the enzyme is purified using affinity chromatography, often facilitated by a His tag. Genetic modifications can further improve enzyme properties. For example, the galactosidase enzyme, LP3485, was cloned and modified to enhance its synthesis capacity. Similarly, the lipase from Geobacillus thermocatenulatus was engineered by removing and reintroducing cysteines for chemical modifications. These processes illustrate the precision required in genetic modification and enzyme production, demonstrating the potential for enhancing enzyme functions through targeted modifications.